Assalamualaikum warahmatullahi wabarakatuh. My name is Haninata Sorwana. So the topic that has been assigned to our group is Crispr-Cas9 in Microbial Research. And we have chosen a journal titled Mobile Crispr-Cas9 Based Anti-Phage System in E. coli. The corresponding author in this journal is Cao Cao. This research are supported by Frontier Science Center for Synthetic Biology and Key Laboratory System, Bioengineering and Collaborative Innovation Center of Chemical Science and Engineering. Both are from University of Tianjin, China. E. coli has been cultivated for the bioproduction of a variety of heterologous proteins, natural metabolites and high-value chemicals, such as to produce isobutanol and ivermectin. Bacteria and natural phages engage in a constant arms race that is driven by a continuous cycle of co-evolution. It promotes the evolution of diverse anti-phage techniques in bacteria as well as corresponding anti-phage strategies in phages. Phages and bacteria interact through absorption, infection, and release. Crispr-Cas9 system offers adaptive immunity by generating memories of previous phage contact, which guide sequence-specific immunity. A mobile Crispr-Cas9 based anti-phage system (MCBAS) for bacteriophages defense in E. coli has been developed. The MCBAS reported here is made up of a Crispr-Cas9 system and a conjugation origin that allows phage sequences to be recognized and delivered into E. coli before or after infection.
Okay, the first step is microbial strain and culture media. There will be plasmid cloning and recipient strain. The plasmid cloning, we derive it from E. coli JM109, and recipient strain, we derive it from E. coli SL1 blue. And then both of the E. coli, we will going to culture it in the Luria bertani media that contain natrium chloride, peptide, and yeast extract. And then this solidified LB media, we're gonna put agar in it. And if necessary, we're gonna put kanamycin, tetracycline, and chloramphenicol. Okay, next we move to the plasmid construction. In plasmid construction, all plasmid is being constructed using standard molecular cloning technique and Gibson assembly. Restriction endonuclease T4 DNA ligase fusion polymerase chain reaction, or we known as PCR, is being used from New England Biolabs. And as the side note, primers is being synthesized by T-Sync and plasmid is being isolated with Tien Prep Mini Plasmid Kits. As the figure below shown, MKR phage is being constructed by inserting red fluorescent protein into M13K07 and PGM0 being constructed by inserting green fluorescent protein, or we known as GFP, and ORIT into BBA K12180111BB.
Third step is conjugation, where E. coli JM109 that contain MCBAS plasmid and E. coli SL1 blue, which is the recipient cells, grown separately in LB medium with appropriate antibiotics. Cells were harvested by centrifugation at force 5000 for 5 minutes, washed twice, and then resuspented in LB medium without antibiotics. Next, equal volume of donor cell and recipient cell were mixed and incubated for 1 hour at 37 degrees Celsius for conjugation. The sample is collected and vortexed for 10 seconds to stop the conjugation and then being diluted. Then the dilution is sprayed onto LB agar that contain tetracycline and chloramphenicol. The plates were incubated overnight at 37 degrees Celsius.
Okay, next method is the preparation of the MKR fish. So what is MKR? MKR is the type of bacterial fish that called as MKR. So for this method, the colonies of E. coli SL1 blue strain that contain the MKR fish was inoculated into the LBK medium and let it grow overnight at 37 degrees Celsius at 220 RPM. Then it was centrifuged at 12,000 RPM for 5 minutes. The supernatant was filtered out, and this filter solution is the prepared MKR fish solution. The assay for the clearing fish DNA in the infected E. coli, we need two types of cell, which are donor cell, which is E. coli JM109, and recipient cell, which is E. coli SL1 blue, were mixed together and incubated in 1 ml of LBCM_TET medium. These cell culture were inoculated for 12 hours at 37 degrees Celsius. After that, 10 microliter of the bacterial culture were removed every 2 hours for the 10-fold dilution assay. This diluted bacterial solution was splitted on the LBCM and LBCM_TET solid medium and were cultured at 37 degrees Celsius. This experiment was repeated for the 3 times. This research continued with the assay for the fish infection prevention, where the colonies of E. coli SL1 blue were selected and inoculated into 1 ml of LBCM-TET medium, and then it mixed together with the MKR fish solution for the infection. This cell culture were inoculated for 12 hours at 37 degrees Celsius at 900 RPM. After that, 10 microliter of the bacterial culture were removed every 2 hours for the 10-fold dilution assay. This diluted bacterial solution was placed on the LBCM and LB_can solid medium and were cultured at 37 degrees Celsius. This experiment was repeated for the 3 times. Okay, the last step for the experiment is when the single colony appear and was count on these 2 types of solid medium, which are LBCM and LBCM_can solid medium. The formula are the colony number on LBCM_can solid medium, which represent the number of E. coli cells that infected by MKR fish, was divided by the colony number on LBCM solid medium, which represent number of all E. coli cells in the diluted bacterial solution, times with 100%. This formula was used to calculate the infection rate in the E. coli cell that caused by the MKR fish.
Next is the benefit of the mobile CAS9 based antiphage system in E. coli, or also known as MCBAS. The first benefit is that it clears fish DNA in E. coli. In order to test the MCBAS antiphage effectiveness, MCBAS were first used to eliminate the DNA of the MKR fish in infected E. coli. As indicated in the picture, the CAS9 system should be transferred from an E. coli donor cell to a recipient cell in order to gradually degrade the DNA of the MKR fish. As shown in this figure, MCBAS contain the transfer ORT, which enable conjugative transfer of MCBAS plasmid from E. coli donor cell to recipient cell. The donor cell carrying PGM0, the recipient cell carrying PUC19 and penicillin resistance, and a mixture of the 2 strains were dropped on LB agars to test the mobile ability of the MCBAS. Only the mixing group showed colonies, suggesting that the MCBAS could be conjugatively transferred from an E. coli donor cell to a recipient cell. This table shows the infection rate of E. coli by incubating the bacterial culture for 12 hours, where the E. coli recipient cells were infected with the MKR fish. And then the E. coli donor cells that contain PGM0, PGM1 and PGM2 were mixed with the recipient cells. As we can see, the infection rate of E. coli carrying PGM1 keep on decreasing until it reached 1.6% after 10 hours of incubation. The final infection rate of PGM2 carrying E. coli was around 0.2%, showing that both PGM1 and PGM2 were efficient at preventing MKR fish replication in vivo. The next benefit is MCBAS can be used to defend against filamentous fish. The effectiveness of MCBAS to defend E. coli from fermented fish were tested using two tests, which are fish DNA clearance test and fish infection prevention test. The first test, fish DNA clearance test, were used to clear fish DNA in infected E. coli. For this test, MCBAS was delivered to infected recipient cells via conjugation to remove the fish DNA. Next is the fish infection prevention test, which MCBAS were used to prevent fish infection in E. coli. The fish was added for infection after delivering MCBAS to uninfected recipient cells via conjugation.
Next is MCBAS can prevent fish infection of E. coli. To test the preventive effect of the MCBAS, E. coli recipient cells that carry PGM0, PGM1 and PGM2 were combined with MKR-PAGE at a ratio of 1 to 500. After incubating the bacterial culture for 12 hours, a PAGE yield experiment was carried out at a regular interval of 2 hours. Based on this graph, E. coli carrying PGM1 had infection rates greater than 2% before 8 hours, 15.2% at 10 hours, and 92% at 12 hours. All infection rates for PGM2 carrying E. coli were below 1%. This result indicate that PGM2 demonstrated a better preventive effect than PGM1. To test if MCBAS works better when there are more MKR-PAGE, more MKR-PAGE were used to infect E. coli with PGM1 and PGM2. When the MOI was 1 to 100, the MKR infection rates of E. coli carrying PGM1 increased to 4% and 99.6% at 6 and 12 hours respectively, while no significant infection was seen in E. coli carrying PGM2. At 1 to 20 MOI, E. coli carrying PGM1 had 1.75% and 100% infection rates at 4 and 12 hours. E. coli carrying PGM2 infection rates increased to 3.8% and 3.7% at 6 and 8 hours, then decreased to 0.6% and 0.1% at 10 and 12 hours. At 1 to 10 MOI, E. coli carrying PGM2 had infection rates of 3.1%, 12.1%, 10.4%, 1.2% and 0.1% at 4, 6, 8, 10 and 12 hours. High concentration of the MKR-PAGE can speed up the infection. When the quantity of MKR-PAGE in the system exceeds PGM2's threshold, E. coli carrying PGM2 infection rate increased at the middle stage, which is at 4, 6, and 8 hours. However, the continuous degradation of PGM2 decreased infection rate over time. In conclusion, MCBAS is developed to defend against bacteriophages in E. coli. By degrading phage DNA in vivo, the MCBAS may effectively rescue E. coli. The enhancement of MCBAS preventative impact can be furthered by adding numerous tRNAs of different targets within phage genome. In establishing microbial cell factories, this approach can ensure a synthetic biological platform for phage defense ability enhancement.